biologists. We are going to talk about inheritance today, and we're going to take some notes, so get your pen and your paper and get ready to write some stuff down. All right, so let's start by thinking about dominant and recessive traits. You already know about that. You know that dominant traits are written with a big letter, a capital letter, and recessive traits are written with a lowercase letter, and that if there is one dominant allele, that's what will be expressed. So we know that. What we want to do now is we want to talk about the other types of inheritance. So the first one we're going to talk about is partial inheritance. This is other. Other types of inheritance. That's our goal for today. Type number 1 is partial dominance. So partial dominance is when you have dominant and recessive traits, but there's not just two options. There's more than two options. The example that you read about in the book was on the K locus. K is the gene name. A locus is the location of the gene. So this is telling you where the location of this gene is. And this gene coded for brindle color. I'm going to say brindle coloration. That's that black and brown stripy pattern that some dogs and some other organisms have. I think about it in dogs. At this location, this K locus, remember that's a location of a gene, we had 3 alleles. Remember, when it's regular Mendelian inheritance, it's usually 2 alleles, a dominant and recessive. In this case, you have 3. The 3 alleles that you read about were K, that's the location, big B, so that seems like it should be dominant. Then you had K, little b, which seems like it should be recessive, but it was little br. And then you had K, Y. So those are my 3 alleles. These 3 alleles coded for different types of coloration. If you had the K big B, it coded for solid black fur. We're talking about dogs. If you had K BR, it coded for brindle pattern. And if you had KY, that coded for yellow fur, because the pigment is not being shown, so it looks yellow to us. Now this is partial dominance because K big B is dominant to KBR, and KBR is dominant to KY, which means that if you have the genotype, I'm going to do this down here, genotype, options. If you had K big B, because you are a diploid individual, you're a dog, or K big B, K little br, that would look like black fur. If you had K little b r, K little br, or K little br, K y, that would be brindle fur. And if you had K y, K y, that was yellow fur. So this is partial dominance because there's a couple things happening, right? The big B for the allele for black fur is dominant to the allele for brindle fur, but the allele for brindle fur is dominant to the allele for yellow fur. So it's partial dominance. Then these are your genotypes, and over here are your phenotypes. Just make sure we have both, genotypes and phenotypes. Another type of inheritance is incomplete dominance. That was partial, now let's look at incomplete. Incomplete meaning like not fully done. So I like to think of incomplete as being an intermediate. That helps me remember it. Incomplete is intermediate. It's somewhere between both my options. Let me show you what I'm talking about. In your book, you read about the KRT71 gene. The KRT71 gene is what coded for different types of keratin. Keratin gene. That keratin is the protein used to make up skin and hair and fingernails or puppy dog nails. And in this case, it was the gene used to determine whether or not your dog had curly fur or straight fur. There were 2 alleles, and the allele options were K for keratin, so we can reuse the same letter, but we're talking about a different allele and a different gene now. And KC was curly fur, like a poodle. Then you had K plus, that is straight fur. It's with a T, right? Yeah, that kind of straight fur. So if you want to write an S, that's okay. Whoever identified this gene did it with a C and a plus. So based on Mendelian genetics, if we had 3 genotypes, genotype options, we would have homozygous dominant, we would have heterozygous, and we would have homozygous recessive. So if this was regular Mendelian genetics, the heterozygous individual would have curly fur, but that's not the case in incomplete dominance. In incomplete dominance, the heterozygous individual has an intermediate fur. So not curly, not straight, instead it's going to be wavy. So homozygous dominant is curly fur. Homozygous recessive is straight fur. Heterozygous is halfway between curly and straight, which is wavy. Think like a labradoodle, like they have that wavy fur. So those are the genotype options, these were the phenotype. So phenotype is what it looks like, genotype is what its allele orientation is. Awesome. We've got another example. Instead of incomplete dominance, it's going to be co-dominance. Co-dominance. So incomplete dominance was in between the two. Think if you had a red flower and a white flower, you make a purple flower. I'm sorry, pink. Red and white make pink. If you had a blue and a red flower, then purple would be intermediate. In codominance, it's going to be both, but not blended together. I'll use the flower example because that makes a little bit more sense in this case. So the gene we're going to be looking at is flower color. We're going to have two alleles. We're going to have a red allele, which is going to be the big R. It is dominant in regular Mendelian genetics, and then we're going to have little r, and that's going to be a white flower. It would be recessive in Mendelian genetics. And in codominance, if I had these options, I would see both of those alleles being expressed. So check it out. Homozygous dominant, you get a red flower. Homozygous recessive, you get a white flower. Heterozygous, you get a red and white flower. Which, have you ever seen that? Would you like to see it? Alright, now you're looking at my computer screen, and we took a red camellia and a white camellia, and we bred them together, and instead of just expressing one of the phenotypes of the parents, you end up with red and white flowers, which is just so cool. Here's another example. In this slide, we'll start here, just scooch over a little bit. This flower, you can see the genus name right there, it's Mirabilis, is often called the 4 o'clock plant because it blooms at 4 o'clock p.m. It's really, really cool. In regular Mendelian genetics, if you had a dominant trait and a recessive trait and you created a heterozygote between these two individuals, the offspring would look like the dominant parent. But in the Mirabilis flower, you can do it in both ways. You can have incomplete dominance, where the red and the white blend together to give you a pink flower, or you can have codominance, where both the red and the white phenotype are expressed. So neither one of those alleles is overpowering the other. Here, this is the same type of flower, the 4 o'clock plant, and you can see the cute pink incomplete dominance, and then you can see this really cool other example of codominance. So the allele from dad and the allele from mom are both being expressed instead of blending together, which they do in incomplete dominance. So now that we know about these other three types of inheritance, let's talk about a cool example that uses at least two of them, and that's going to be human blood. So exciting. Don't worry, I'm not going to show you blood or needles, we're just going to talk about it. So human blood uses the ABO blood typing system. Which is really cool, and the ABO blood typing system is based on 3 alleles. The 3 alleles are, they're written kind of weird, so get ready. You have capital I, capital A, capital I, capital B, and then little I. Capital IA codes for A antigens. Now, what's an antigen? An antigen is a little protein on the outside of your red blood cell. So here is my red blood cell. It's circular. It's kind of got a divot in the center. So imagine that's like a pushed in little divot. It's called a biconcave disc. It's really cool. And if you have A antigens, you have these little proteins sticking out the side that we called A proteins. So they are A antigens. If you have capital I, capital B, you have B antigens. So that would be like this. This is your red blood cell, and then sticking out of it, you have B proteins, B antigens. So these little things sticking out of your cell are antigens. Antigen. Pretty neat, right? Little I, that codes for no antigen. This is not a bad thing. This is a really common thing. That just means that your red blood cell sits there. Just no little protein sticking out it. It still does its job. There's nothing wrong with it. Just no antigens. In fact, not having antigens is really common in blood types. So we have 3 alleles. Let's talk about the genotypes and phenotypes. The genotypes are either going to give you A blood, B blood, or O blood, or a fun 4th option. So if you have 2 of the A antigen allele, you have A blood. But A is dominant to no antigen, so you can be 2 of those, or 1 A antigen allele and 1 no antigen allele, and either way you would have A blood. Because A is dominant to no antigen, your red blood cell would look like this one. You would have A proteins on the outside of your red blood cell. Same thing for B blood. B blood can be 2 copies of the allele that gives you B antigens, or 1 copy of it and 1 copy of the allele that gives you no antigens, and you would have B blood. So that would be this one right here. So A is dominant to I, B is dominant to I, little I, and if you had little I, you would have O blood. O type blood is just a red blood cell with no antigens on the outside. So you may be wondering, what about AB blood? An individual with the allele for A antigens and B antigens would be AB blood. That would look like this. You have your red blood cell, and you have A's and B's. 2 types of antigens. So 3 alleles, we have 6 genotypes, and we have 4 outcomes. Since both A and B are being expressed, this is an example of codominance. And since A and B are both dominant to I, but not dominant to each other, this is partial dominance. Partial dominance. So we have an example of codominance and partial dominance. You may be wondering, what about positive and negative? Because, right, I have A negative blood. What does that negative mean? That refers to another allele. And this time it's called the RH allele. The RH allele is also sometimes called the RH factor, and it codes for 49 different types of antigens that could be on the outside of your red blood cell. So it's really way more complicated than we ever talk about. We pretend that you either have it or you don't, and that it follows basic inheritance. So if you had 2 copies of the dominant, or if you were heterozygous, you would be positive blood. Yep. And if you were homozygous recessive, you would be negative. So that's a simplification, but that's what we roll with. So if I have A negative blood, which I do, that means that my genotype is AA or A little I, depends on what my parents are. I happen to know that my mom has O blood, so I must be A little I because my mom only has little i's to give me. And since I'm negative, I also have little r. So that is my genotype, and my phenotype is A negative blood. Pretty neat, right? You know your blood type. If you don't, you can donate blood if you fit into their guidelines of who's allowed to donate blood, which are a little bit biased, and we can talk about that in person. But if you can, donate blood and they will tell you your blood type. And donating blood is really important because it saves lives every day. So if that's something that you're willing to do, go ahead and do it. I'm a big fan of donating blood. So I do it anytime I can, even though I'm totally a fainter, but that's okay. It's worth it. Hope you have a great rest of your day. Email me with questions and we can totally talk about blood typing more during class. Bye.